Determination of the precise order of nucleotides within a DNA molecule is popularly known as DNA sequencing. About 4 decades ago, in the year 1977, Sanger and Max and Gilbert made a breakthrough that revolutionized the world of biological science by sequencing the bacteriophage. In the article, they describe the process of this way. The single-strand DNA is clipped at the specific location with the help of the chemical at the specific base location, and then fragments of DNA are then run on polyacrylamide gel. This process consists on the next steps. A single or double-strand DNA fragment is labeled at the 5' or 3' ends of one or both strands with phosphorus 32. The purines, adenine, and guanine are cutting a dead formic acid to the DNA fragment labeled to break the link between a purine, A or G, and the deoxyribose to which it is attached, and so they can have different apurinic sites. The guanines are methylated with dimethyl sulfate, DMS. The guanine is removed from the polynucleotide chain. The reaction for cutting the pyrimides uses the hydrazine reagents, which cut the base cytosine and thymine. The presence of sodium chloride inhibits the reaction of hydrazine with thymine and produces only cytosine-terminating fragments. To complete the before reaction, they added piperidine. The reaction were loaded on high percentage polyacrylamide gels and the fragment resolved by electrophoresis. The gel was transferred to a light-proof X-ray film cassette. A piece of X-ray film placed over the gel and the cassette placed in a freezer for several days. The process involved interpreting the binding pattern relative to the full chemical reaction.
2009, Markham-Gilbert sequencing method was applied to determine thrombin-binding aptamer sequences that have isoguanine, IG, for possible alternative use as an antithrombotic agent. Aptamers are sequences of single-stranded nucleic acids isolated from oligonucleotide libraries determined by in vitro means. It was found that such thrombin aptamers with more IG have greater ability to bind with alpha-thrombin, which is a coagulant. These aptamer sequences were mainly subjected to G reaction as well as G plus IG reaction to be finally analyzed by phage electrophoresis. In 2005, Max and Gilbert method was performed to identify a relationship between fascicularin and cell lines efficient in DNA repair. Fascicularin is a compound, an alkaloid containing thiocyanate, and is obtained from acidia that shows cytotoxic properties. This sequencing analysis was performed on duplex DNA treated with 32P and fascicularin. They used A plus G reaction and G reaction for subsequent gel analysis. Finally, fascicularin was found to have mechanisms similar to anti-cancer drugs such as meclotheriramine.
The enzymatic method for DNA sequencing is also known as Sanger sequencing, after its developer Frederick Sanger, who first described the method in 1977. The result of the enzymatic method is very similar to the chemical method, but the procedure is completely different. First step, a polymerase chain reaction, PCR, with fluorescent chain-terminating dd-NTPs. First, a primer is transferred to 4 tubes. Then, DNA polymerase is added to each tube. Subsequently, dNTPs are added to each tube, and then specific dd-NTPs, dideoxynucleotides, which can be guanine, adenine, cytosine, or thymine, are put on each tube. Then, the tubes are collocated in the thermocycler, and an N-chain PCR is performed. Each tube has sequences that end with a specific nucleotide, depending on the dd-NTP added. Second step, site preparation by gel electrophoresis. On the next step, gel electrophoresis is performed, putting each sample containing different dd-NTPs in separate lanes. Due to the fact that DNA is negatively charged, the molecules will travel to the positively charged region, and the heavier DNA fragments will take longer to travel along the gel. Third step, gel analysis and determination of DNA sequence. It is important to label whether a primer or a dNTP with radioactive phosphorus, 32P, in order to be able to see the DNA bands in the X-ray film. Overnight, the DNA fragments will stick to the X-ray film, and the fragments will be visible in the sequence ready to read. Finally, it's worth mentioning that Sanger sequencing can be conducted whether in manual form or in an automated manner.
In 2015, some students from the Applied and Biomedical Sciences School in Australia did a practice through the search sequencing method, looking and creating their primers to be able to find the complete genome of influenza A virus, AIV, and determine if resorting has occurred or if any other pathogenic traits were present. However, after a long experimentation, they were not able to design a set of primers that could universally amplify the 6 internal genes from influenza A subtypes. Their experiment was functional to 3 subtype viruses that they chose, proving that search sequencing method was useful, but influenza A virus had too many variants for them to find all the primers needed for the sequencing. In 2017, a group of scientists were looking for a method to identify Cas9-induced friendship that cause friendship indel mutation and disrupt protein expressions. Therefore, they create a simple and efficient method to identify friendship mutations in diploid genomes, but based on Sanger sequencing method and MS worldwide call search. With these methods, they could predict exactly how many nucleotides were removed or inserted into each allele of the genome.
There are several equipment developed by Thermo Fisher Scientific Company for the Sanger method. The 3 more relevant equipment are the SIG Studio Genetic Analyzer, the 3500 series genetic analyzer, and the Refresh 3730 series genetic analyzer. Being the latest, the more technologically advanced due to the following reasons. The Refresh 3730 series genetic analyzer can have 48 capillaries on the standard version, or 96 capillaries in the XL version. It fits up to 16 samples and has an integrated plate stacker. The minimum run time of this piece of equipment is 20 minutes, and it can read sequences up to 900 base pairs. Nevertheless, the rate of base pair reads per day can be up to 1.38 million on the standard version, or even 2.76 million on the XL version. This piece of equipment played a key role in a 2020 study developed by Ha et al., where their objective was to study the genetic diversity in Pangasius spp, collected in Bangladesh, based on mitochondrial cytochrome B gene sequence analysis. They used the Refresh 3730 series genetic analyzer in order to do the sequencing of the cytochrome B gene fragment.
Today, the most widely used method for nucleotide acid sequencing is Sanger method. However, it's fair to say that Maxam and Gilbert method is best suited for determining the sequence of a short DNA fragment because it can determine the sequence from first base. In contrast, the Sanger method only allows reading from the base 10 to 20. These methods have been the base from which researchers and scientists across the various fields are utilizing these technologies from the variety of applications, including food security, by developing berry crops and crop yields, livestock, improved diagnostics, prognostics, and therapies for many complex diseases.